Hello, everyone. So my name is Max, and today I'm going to be presenting on the realm of biochemistry. So my project title is Investigating the Target Protein, DNA2, in the Cytosolic iron-sulfur cluster assembly pathway. To begin, iron-sulfur clusters are an essential component of all living organisms. So these compounds contain iron groups, which are surrounded by sulfur, allowing them to hold charges and electrons. So basically, iron sulfur clusters move all throughout our body, and they kind of transport the energy that we receive from eating foods. So our metabolism creates energy, and that energy from the mitochondria needs to be moved around all of the different cells in our body. So without iron sulfur clusters, life would not function, and most of the proteins in our body would not really be able to fold correctly, leading to endless problems. So the SIA pathway, shown on the left of the slide, is the way in which iron sulfur clusters are created, distributed, and eventually inserted into a target protein. So this is very similar to Amazon in the fact that something can be created, sent to a distribution center, and then finally delivered to like your house or something like that. So in this case, the delivery would be an apoprotein, so a little bit smaller scale, I would say. But yeah, so proteins only become active once this iron sulfur cluster is inserted into them. Um, and there are many steps in the SIA pathway. But my project is focusing solely on the target protein DNA2, which is going to be the last stage where the iron sulfur cluster is delivered or inserted into the protein. This protein is shown on the right in this kind of globular structure, and it needs an iron sulfur cluster in order to be active in DNA transcription and replication, which are two vital processes in all life. So, in order for me to study the SIA pathway as well as these processes further, we need to first purify DNA2, which became a very difficult task, as you'll see. So, with this in mind, my lab is currently trying to uncover the mysteries behind the SIA pathway. So, members of my lab previously have uncovered that the majority of apoproteins that receive this iron sulfur cluster contain an aromatic tryptophan residue at the end of their amino acid sequence. So, basically, this kind of just means that there's like a little W at the end of one of the amino acid sequences. So, this is very interesting. However, we've noticed that some proteins do not have this aromatic residue. So DNA2 is very unique in the fact that it does not. However, it still binds to the SIA pathway. So we really would like to understand how this works. DNA2 needs to be purified in order to do this first. So my project was to devise a plan to purify DNA2 and then hopefully in the future run some assays to test it in the SIA pathway. So traditional methods of purification that my lab normally used failed for this specific protein. So we had to develop some new in tandem purification sequence. So I will discuss IPTG growth conditions, affinity chromatography, and then finally a cation exchange chromatography, which are the three methods that I use to fully purify DNA2. The first step was to determine the ideal growing conditions to best express DNA2. So, in summary, DNA2 protein is grown in a non-pathogenic strain of E. coli cells, but I can add different reagents to the media to help better express the protein. In the end, we want to have the highest concentration of DNA2 protein in order to perform these experiments that we're trying to test with the SIA pathway. So I added IPTG, iron, cysteine, and then I also tried an autoinduction broth to determine which method would produce the highest concentration of DNA2. I found that whenever I add IPTG to the growth of DNA2, the highest amount of DNA2 was recorded. So this can be shown with the SDS page gel, which is on the right, which is basically a way to kind of take a picture, take a snapshot of like proteins in a solution. So each one of these bands on this gel is a protein. And at the top where you can see the red line with DNA2, the very top band, that's the protein that I'm looking for. So you can see here that in IPTG, which is in the first column, it has the darkest band, meaning that IPTG was the most successful at making the most DNA to protein. So in all future future studies, I'll be using this to purify the protein. Once the cells are grown, we must chemically cut them open to get our protein of interest. So when we cut open E. coli cells to extract the protein, there are thousands of extraneous proteins that just naturally are in a cell. Um, so we must use specific techniques to get out just the protein of interest, DNA2 for this case. To do this well, we use something called affinity chromatography. The basis of this technique is that we have a protein and we put an artificial tag on the end of it, and it's kind of like Velcro in a sense. So we have this resin that has one side of the Velcro, and then my specific protein has a different side of it. So whenever I put my protein solution onto this column, my protein sticks to the Velcro just like a normal piece of Velcro. However, the issue is sometimes, as we know in Velcro, there can be some impurities, maybe like some hair or something that kind of gets stuck in the Velcro as well. And this can happen in affinity chromatography as well, leading to different proteins that we don't want also being purified, quote unquote. So the mechanism here is a bit complex, but in the end, we should be able to see a purified DNA2 protein in the pink sample all the way on the right. Usually this technique is all we really need to do to purify a protein. However, with DNA2, this was not the case. So upon completion of the affinity chromatography, another SDS page gel was run to see if the method was successful. On the left side of the gel, you can see the mess of the thousands of proteins that are normally in a cell in the initial solution. And then on the right, you can see the results of my affinity chromatography experiment. So in red, I have DNA2, and then in green, I have impurities. So these impurities are basically just other proteins that can kind of get in the way. So I want to make sure that I only have DNA2 rather than these other proteins, as my assays in the future could be influenced by the others. So after the experiment, there were still a few impurities that I must remove, but the solution is definitely much cleaner, as you can see from the initial sample. We have about 10 impurities rather than 10,000. So finally, after many, many unsuccessful experiments, I tried cation exchange chromatography to completely purify DNA2. This method is pretty complex, but in the end, it's basically just a purification using electrostatics. So at a pH of 8, DNA2 is a highly positively charged protein. Basically, I can use this to my advantage. So I can set the pH of my solution to 8, have DNA2 be very positively charged, and then run it on a resin very similar to affinity chromatography, except this time the resin doesn't have Velcro tag, it has a negative charge attached to it. So this will allow my protein to stick to the resin, and all the other proteins shouldn't be necessarily positive, although it could be, but in my case, luckily it was not. We can then put like a non-ionic solution to help elute this protein off. And in the end, you can see on the right side of the gel that this method was successful in fully purifying the protein, whereas there's only one band on the gel, meaning there's no impurities. So before the experiment, I had about 10, and after, there are none, meaning cation exchange was the final piece of my puzzle to fully purify the protein. In summary, over the last year, I tried many different techniques to purify DNA2. Many of these failed, but in the end, the following procedure was successful. First, I would induce my growth with IPTG. Then I would use affinity chromatography to help purify at least some of the DNA2 and get rid of most of the impurities. And lastly, I would run that sample on a cation exchange, which then would lead to just the DNA2 protein. Now that DNA2 is fully purified, future research can be conducted to analyze its mechanism in the CIA pathway. So we can run some different assays to figure out how does a non-tryptophan, a protein that does not have an N-terminus with a tryptophan residue, how can this function in the CIA pathway? And so that will help unveil all of the mechanisms within DNA transcription and replication, as I mentioned earlier, as well as the CIA pathway in the end. So overall, all of this research can be applied to metabolic diseases, since iron sulfur clusters are very essential in all of our metabolic processes. And so in conclusion, I just want to thank the Pearlstein Lab Group for their help on this project, as I definitely needed it. and as well as the Kilachand faculty, administration, and all of my advisors. So, thanks. And I can take some questions if anyone has any.
Hi, I wanted to thank you for your presentation. It was really interesting. I just had a quick question about the costs of doing some of these experiments. I know that BU obviously has funding, so you were able to do these. But could these, like, if you think you had better technology or more expensive equipment, do you think you would have had better results, or it's just a problem with this protein and it's a tricky one?
So there's definitely other ways to purify proteins that can be much more, I guess, expensive. This was a pretty expensive project in itself, with some of these resins costing thousands of dollars a piece. Um, but you really have to kind of learn about the protein and its optimal pH and just a lot more about it to understand which method that you should try first. So since this was still like a early on application for this specific protein, we tested it with our traditional ways. They didn't work. So we just tried some other ways that we kind of had around the lab. And that's ended up working successfully. So yeah.
Max, I'm going to try not to cry. I'm so proud of you, and it's so cool to see you 4 years later standing up here finishing your final project. So, I have a comment, not a question. When I took chemistry, the last time I took chemistry was in high school, and I got a D, and it was a Mercy D. I learned more from listening to you just now than I have in a very, very long time about chemistry. And so I was wondering if you had ever thought about being a chemistry teacher.
You know, I actually have thought about that before, especially in high school. I had a really, really good chemistry teacher. And that's kind of like why I'm studying this today. And so I thought about doing it because I really like teaching people about the things that I love. But I don't really know if I would be able to do like a classroom of like high school or students. That might be a lot, but stay tuned. It's possible. So, yeah.
Thanks, Max. This was really got to learn a lot. So my office is right next to where Max has been doing all this lab, and I've been seeing him coming and going and wondering what he was doing. So this was great. So two chemistry-driven questions. So first of all, is it known that the tryptophan in the other non-DNA2 proteins is binding, or is that just an empirical observation and the function hasn't been definitively identified?
So this is actually known. So my, I guess, sophomore year advisor in the lab, Melissa Marquez, she did this as her PhD thesis. And so she completely, so basically this is a known fact now that tryptophan is what's causing this binding. We've done some assays where we remove the tryptophan and then the protein no longer binds. And so we found that just this tryptophan is necessary in order to promote binding. And so it's very interesting that some proteins, they solely rely on the tryptophan, whereas DNA2 doesn't even have a tryptophan in the C terminus like most of the proteins. Yeah.
Okay, so then wonderful lead into my second question. What is special about DNA2? So is it sulfur rich, got a lot of carboxylates, I'm surprised that you quote your pH to only one sig fig. I mean, 7.9 versus 8.1. What do you know about DNA2 now? I'm just curious.
So DNA2 is one, it's an extremely large protein. So the most important thing or the thing that I think is kind of leading this binding is that it's so large that it can alter like its conformation with this iron sulfur cluster a lot. So we think it can possibly just bind in different locations. So whereas like a lot of the ones with the tryptophan residue, they bind specifically to CA2, which is one of our proteins in this like CA targeting complex. Whereas DNA2, it's been proven that it binds to MET18 and potentially CA2. So we've been able to demonstrate that DNA2 binds to the full targeting complex with MET18, CA1, and CA2, but we haven't really been able to do any assays, since I kind of ran out of time, to see if DNA2 binds to solely MET18, solely CA2, or solely CA1. So we kind of think it has something to do with its difference in binding in the C targeting complex. But the pH, 8 has worked pretty well. So typically, the protein's PI is 6.8. So we want to have a pH that's much higher or much lower. And if it was too low, that would be very acidic. So we don't want to do that. So 8 has kind of worked well. 8.1 has also worked pretty well. Just kind of around there, we kind of went to a flat number, and it's worked for us since, so we don't really want to change it too much. But we did have an issue with our pH meter this past semester. So 7.9, 8, 8.1 could have been anywhere around there that it's been working. So I think something like that would work, yeah. But any other questions?
Thank you so much, Max.
Cool.